Thank you, Alan. It gives me great personal pleasure also to introduce Bill formally to you as the first speaker for this morning. Dr. Crowley is professor of medicine and at the Harvard Medical School and director of its Center of Excellence in Reproductive Endocrinology. Bill has served as the director of clinical research at the Massachusetts General Hospital for the past 13 years and is also the founder and current president of the national organization known as the Clinical Research Forum. Over 10 years ago, when Bill assembled a founding group to create the forum, and I'm proud to say the Einstein Dean was a member of that, it was already clear that he would be a powerful voice, Bill, would be a powerful voice for clinical research in the U.S. Under his stewardship, excuse me, the forum has grown to be an organization of 65 of the leading academic medical centers, and Bill reminds us that they constitute over 75% of the total extramural NIH budget. Bill was also a member of the Institute of Medicine's Roundtable, where he co-authored the classic papers in the Journal of the American Medical Association outlining the challenges and possible solutions going forward for clinical research. Those of you who are arriving late, there are seats in the middle. During his 40 years at the MGH, Bill pioneered the use of gonadotropin-releasing hormone for the treatment of precocious puberty in children. This is now also used to treat prostate cancer, endometriosis, uterine fibroids, and represents a $2 billion pharmaceutical market. He also went on to develop the use of pulsatile GnRH to treat infertility, and his laboratory is now identifying the roles of genes responsible for puberty in the GnRH deficient human model. Among his honors, Bill received both the NIH's Clinical Research Center National Award and the Endocrine Society Award for Excellence. He was named an honorary fellow in the Royal Society of Medicine, Ireland, and was the first male to receive the Mentor of the Year award from Women in Endocrinology. A signal honor. Dr. Crowley served as president of the Endocrine Society and has also received its highest honor, the Koch Medal, and in 2007 he was awarded the Fondation Ipsen Award for Endocrine Research, an international prize in endocrinology. Bill, it's an honor and a pleasure to welcome you to the Bronx.
Well, I'd like to begin by thanking Christine and Stacy for the wonderful hospitality and arranging things, and particularly for Harry, Paul, and Brian. Congratulations on getting a CTSC. Well deserved on your part. I think it's an exciting and marker of the future, I think. Great. In addition, I can't help but comment about how lucky you are to have your current dean. I've known Alan since he was a medical student, and certainly as a director of NIDDK, was one of the most distinguished and far-reaching in terms of his thinking directors at the NIH, and I think it's exceptionally lucky recruitment of your part, and I can already see the changes in 2 years. So Einstein is one of the places that's really equipped to go forward into the future in the sort of 21st century, which will be the century of biology, no question, following the century of chemistry and physics. I think what I wanted to do actually is to drop back here and give you a little bit of an overview of where I think the changing models are occurring, because it's important to understand when you're in an interregnum. And interregnums are particularly tough for young investigators because the rules are changing, but it's hard for older investigators as well. And I think this is one of those times where tectonic shifts are occurring, enabling tools are appearing, rules are changing, and the CTSA is but one epiphenomenon of that. So what I'm going to try to do is to have some look at the implications of this across scientific areas. Really, like many things, this is a story of two men, actually, and their models. The first is Fuller Albright, who's the father of modern endocrinology, the ultimate bench-to-bedside physician. He practiced in what I call the aquatic era of endocrinology. That was when we were measuring everything in the urine. And because of the lack of powerful tools, he was able to describe things at the bedside. And I think, as I'll try to make the case, we're coming back to an era when that's important. A counterpart of his was Vannevar Bush, Both of these men were born in the late 19th century. Both were contemporary pioneers in the 20th century Boston science. Vannevar Bush had been the provost of MIT. He's an electrical engineer, and actually wrote a famous paper, as we think, that anticipated the internet. Although that's not why I'm citing him today, because it's another government report called Science, the Endless Frontier that's the topic of today. They died in a relatively short time period of, actually Phil Robight died when I was in my internship in 1969, and Vannevar Bush died 5 years later. They were both thought leaders with major impacts on science policy, and I think the themes that interwove between these two men are playing themselves out today still. Interestingly, they both used arrow diagrams. Now, as all of you know, if you take an arrow away from an endocrinologist, he can't talk. So, in fact, Vannevar Bush also used arrow diagrams, so I thought that's just a little historical vignette. So Albright, Albright put in place a very important historical trend, which was bedside observation, in which he would attempt to bring things back to the laboratory. Unfortunately, the tools were not precise, they were not accurate, most of them were done in the urine, and from the 30s to the 60s, this was where he describes 7 or 9 syndromes, depending upon how you want to count them, but the feedback from the bench to the bedside was small. In 1945, a signal event happened. Franklin Delano Roosevelt asked Bush to take a look at science in America after World War II. And he wrote a famous government report called Science, the Endless Frontier. In that report, he made the observation that science should be open and available, this being very different from the way it was conducted during the defense era. He also made a very important policy point that the government should be the funder of this. And this established the foundation for subsequent NIH funding. And over the next 10 years, the NIH was set up. And this changed completely academic health centers. At the turn of the last century, you went to an academic health center, like the Massachusetts General Hospital, to die if you were poor. Because if you were wealthy, you had your physician attending you at home. So they were almshouses, was what they were known. And over this century, because of NIH funding largely, these became the prestigious institutions that you see Einstein looking like today. He also established a very linear model of research, starting with basic research and moving forward, saying that basic research was the cornerstone of everything. You then try things in animal models, and if in fact you had any important proof of principle, you could go on and test with humans, but that was sort of just to prove it was important. Now, if you really look at how did academics respond to this, they built the infrastructure that was appropriate for the greatest leverage investment. That is, they put in basic research laboratories, they subsidized them, the medical schools hired people, and we'll go through the policy implications of this. But the infrastructure that was for testing in humans was largely pharmaceutical companies. And, by the way, viewed as competitive, so it would be removed every time they were done. So the policy consequences of this report were quite significant of the Bush doctrine. No pun intended. I couldn't. Yeah, it's apolitical. So the question is, who are you going to recruit? Who were you going to make your investments? Who are you going to retain? And I'm going to play this out through each of these areas. Well, this was a no-brainer for the NIH. They were going to spend the majority of their money on basic research. It's been at least 2/3, depending upon how you want to count and categorize it. No two institutes, by the way, define clinical research the same, so it's hard to get objective data. But the closest data you can get is about 2-to-1. There are other estimates which are a little more optimistic, but having been on the director's panel and looked at this and then seeing at the IOM, it's about 2-to-1. The medical schools, this was quite easy. You basically hired basic science faculty to teach your medical student. You geared your promotions committee for their rapid movement. You allocated space and you subsidized their core laboratories. And you basically became a PhD place exclusively for the first 2 years in medical school. Now, the academic centers, or the affiliates as they were called, They really took a page right out of the medical schools. They really gave a lot of space to basic research. They decided that all the core laboratories for those people had to be subsidized, and that the future clinical investigators would be the MD PhDs that went into the MST programs. They were the future investment for translational vehicles. P.S. A corollary to this is if you wanted to do clinical research, you had to have industrial money and you were to be a profit center. That is, there was no subsidization. You were supposed to bring in revenues. You had 25% indirect cost, and that was revenues in excess of expenditures. Being a not-for-profit, we never say the P word, but it's revenues in excess of expenditures. Now, what were you going to do about clinical research? Well, the NIH had the intramural programs, which in my era, I missed those. I actually got drafted in 1969. But everybody at the MGH, plus or minus 20 years, went to the NIH, actually, as they were called the Yellow Berets. The idea of getting around the war in Vietnam was that you went there. But this drew the best and brightest. You know, having a carrot stick is a good way to do it, the carrot being the NIH science, the stick being you could avoid the draft if you went there. And for decades, the NIH drew the cream of the crop at the NIH. And this really fueled all of the science programs of the 60s, 70s, and 80s. The medical schools just took a pass on clinical research. They sent people out to the hospitals, and they didn't really make any investment on those. In terms of the academic health centers, that was largely industrial academic relations and pharma trials. And that was really got so severe that actually that's where the GCRC programs came from, that there had to be something else other than pharma trials. In terms of populations, which you'll hear from Nancy Bennett and other people this afternoon, the NIH looked at this from the point of view of rare populations. They had money to import people, and in fact, they studied leprechauns for a long time. It was an important form of insulin resistance, but you know, eventually you do run out of leprechauns, no matter what pot of gold you have at the end of their insulin resistance. The rare patient populations that you have are actually very important and prismatic, actually. So they really did, with the fact that they had bright young people and terrific staff, Jesse Roth, the people that were there, just wonderful. The medical schools, again, took a pass on the population issue, hadn't discovered epidemiology, and left it to the academic centers, where, in fact, industry loomed large again, and the sort of proof of principle concept was done there. Now, the GCRCs came later because this became so severe, it became clear to the NIH that there had to be some other way of leveraging human research, and the laboratories of human research were put in place by the CRR, the Center for Research Resources, which grew into the GCRC program. So really what happened in the Bush model is this all got reversed, that the bench power of molecular biology, its speed, its swiftness, its ability to tell stories completely was the driving force for research in the last 50 years. And in fact, what happened as a law of unintended consequences, clinical observation really began to shrink in importance everywhere in these places. And anytime a dominant tool comes in place and there's a model, this changes the equilibrium. Now, what were the lasting policy implications that we live with today? The first was the rise of the basic research model. Any physician during my era in 1969 is you were advised, get away from the distractions of hospitals. Go to an institute, whether it was the Rockefeller or the Salk or MIT or the NIH. Seek to get a basic program going in those laboratories. Free yourself from teaching and patient care commitments. They were dilutional, and they weren't really scientific endeavors because of their lack of precision. The promotions committee facilitated the rise of these sorts of people, and the old model of a PhD, you have to be a first author, then you must be a senior author, and your mentor must disappear, was the dominant model, and still is the dominant model at Harvard. Teams were viewed with great suspicion, and in fact it was dilutive and it was orchestrated in some way. Industrial funding was viewed in a similar way, and if you weren't able to get NIH funds, you weren't going to get promoted. The second model, which persists today, is that clinical researchers became thought of in shorthand as second-class scientists. And the reason is first of all, it was left to the hospitals, and it was not the medical school's responsibility, largely pharmaceutical in origin, and occasionally people would go into a basic lab, find it not to their liking, go back to the hospitals and have long careers. The basic scientists viewed these guys as second-class citizens and didn't recognize some of them were very smart guys, just didn't fit their particular model, but they viewed them as washouts. And that's still present in the older faculty members, particularly basic scientists. As I see young faculty members now realizing the mutations and the pathophysiology at the hospitals, they're changing their mind and starting to come out and seek. And I see this as Director of Clinical Research at the MGH. We have a budget of $528 million. 75% of that's basic research. And my most common call is, where can I find the guy who takes care of these particular patients from the basic scientist trying to build teams? So the impetus has come from the basic scientist, but there is this leftover feeling of clinical researchers being second class. And the abiding infrastructure that we were left with here is that basic research cores are subsidized, clinical research cores must be profit centers as the corollary. Now, it's very clear that the industry views this as competitive. And this is a big deal for industry. If you can view something as pre-competitive, like the way the NIH Foundation has now sold proteomics to pharma, that the more proteomics we know, the better off everybody will be, they will invest in it. If, on the other hand, it gets the label competitive on it, they won't invest in it at all. In fact, when an industrial company comes in and does a clinical trial, when they leave, if they could roll up the sidewalks and take it with them, they would do that, because why would you leave a sidewalk? It's the next guy will use it. And as a result, competitive thinking dominated the building of infrastructure. And to this day, pharma does not want to build infrastructure at academic centers. And when you peel away why, it's money, it's expense, it's timing, it's the competitive trail that is left for somebody else. So as a result, the NIH had to step in here, and that's where the GCRCs began to initially subsidize them. Now, what is the abiding implication of this? Well, actually, drug development follows this stepwise process slavishly. And this is the consequence of Vannevar Bush. Target ID, chemistry, in vitro studies, animal tox, and then a series of clinical trials if you have something worthwhile. Now, what are the consequences of this? Well, this is slow. It takes at least 10 years to develop an average drug. It's expensive. It's $100 million a drug. But that belies the true expense, because 9 out of 10 drugs fail, and most fail in late clinical trials, half because of lack of efficacy and half because of safety. So the average cost is $1 billion a drug. Well, we just simply can't afford $1 billion a drug in our society. We can afford $850 billion to bail out banks, but we can't afford $1 billion a drug. And in addition to this, there's a low record of new molecular entities. The lowest in decades has occurred in the last several years. The driver, of course, is the regulatory process and the linear model that is linear, unidirectional, and conforms to the Bush model. And the problem, they gain access to patients too late. They haven't used humanized targets. They can't predict the toxicity. And suddenly, when they get into large population trials, mirabile dictu, there is a huge amount of stuff that they haven't seen, because they screened it in relation to one target only, and off-target sites, both good and bad, statins good, Vioxx bad, are the things that emerge in large-scale clinical trials that could have been predicted if they had had a different model of development. So there are 4 consequences of this. There is impressive loss of investor value in pharma company. From 2000 to 2007, the top 16 pharma companies lost $850 billion. Now, I used to have trouble getting people's head around $850 billion. How do you contextualize $850 billion? Well, we now have a way of contextualizing it. And unfortunately, this is by a report from J.P. Garnier in Harvard Business Review, who's the CEO of one of these companies, and I'll show you the data. If you look at this from the pharmaceutical years 1985 to 2000 and the gradual increase, what you look at there is the loss in 7 years. And now that looks like a small loss, but that's $850 billion of non-gain. So they are in trouble. They understand their business model is in trouble. If you talk to them quite privately rather than as a group where they respond differently, they understand they're seeking another model, and they don't know what the model is. None of them do. That's the other problem. Of course, the R&D costs are rising, and their new chemical entities are declining. And that's shown in this graph here from Nature this year that shows the R&D expenditures rising, the new molecular entity output. This is the graph of trouble, you see. And finally, there is this lack of innovation and this sort of, really, they're having the bench-to-bedside problem. They call it the valley of death concept, which is that the pipelines from a basic science to moving into the human is broken down. They put the link in the sausage between research and development. That's how they call it. But in fact, there's a gap there. And in fact, this is a picture of the gap. You see the basic scientist there, the clinician on the other side, and the patient death in the middle. The other thing is, and talk about a driver here, as if this isn't worrisome enough, 50% of all medications that are on the market now in pharma are coming off patent by 2012. Boy, if there isn't a setting to work with industry and academics, I've never seen it but this list here. And the CTSAs and the kind of investments you're making wisely really position you extremely well with your patient populations to be able to deal with some of these things. This, by the way, is their new model, and the big deal is biomarkers. They want to have a biomarker development so they don't have to spend all this money in clinical research. And I think that's one of the solutions, but I think far more important is retooling the model, humanizing the system. There's a lot to be fixed here, I think. But many of the answers lie within the academic community, and they understand that. So what's the big change? This is a gloomy, presentation if you really look at it. The thing is going to hell in a handbag here. You know, where's the opportunity if you're a young person here? Don't leave. There's another half of this talk. The emergence of these powerful new precise tools for clinical research, the imaging tools, the bioengineering, the human genome projects, and all its derivative omic technologies with the IT revolution that's empowering these things to look at massive amounts of data quickly, efficiently, is changing all of this. There were 3 signal events, if you were watching, that were paradigm shifters. They all came from neurology. The first was Huntington's disease, when Jim Gazella used linkage for the first time to link a disease with a gene, and that was Huntington's disease. Now, he had a technician, Rudy Tanzi, who's now become a famous neuroscientist on his own, but he was a technician at that point. And Jim had come up with this very complex family. He thought linkage with these anonymous markers would be kind of cool, and he gave it to his technician to run all the family's anonymous markers and to look at them. And the first gel Rudy Tanzi ever ran in his life as a technician linked Huntington's disease to disease. Now talk about a charmed life. So now that we know the whole genome, Rudy's gone back and calculated, it's a tandem repeat, how many there are in the human genome, which we now know. And basically, the chance of his first gel doing this, even if you knew everything, was 1 in 35,000. In other words, the same frequency of winning the Irish sweepstakes was on the first gel. So he's actually now gotten his PhD and is doing a lot of work on Huntington's disease, which we'll get to in a moment. ALS, Bob Brown linked another family to superoxide dismutase. Now, Bob's an MD, PhD, very smart guy. He never heard of superoxide dismutase. Had to go to the Merck manual to look it up to see what it did. The idea that it was in lower motor neurons and in charge of the apoptosis pathway, nobody knew. And that was the point, because the Alzheimer's disease also took advantage of the pathology, the linkage, the Down syndrome markers as a candidate gene in presenilin. Now, what these 3 diseases showed is that all the neuroscientists and all the King's men were working on nothing related to any of these 3 genes. So that had to be a signal event that everybody was wrong in the neuroscience community. And this new tool was a way to look at this. So it's clear to recognize a paradigm shift if you're a young person when you see it. These 3 observations were rule changers in the 90s. It meant that the answer is in the patients. And the methods were now available, unlike at the time of Fuller Albright, to cull those things out. And there's the opportunity for young people. Diagnostically, the molecular markers, biomarkers, are clearly going to be keys to the future of this and drug responses, and they're all really in the Human Genome Project. So the concept of a tipping point occurs because these technologies are important. The NIH programs on clinical research, the director's panel that I was on, the appearance of K23s, K24s, the clinical research loan repayment. Now, I'll tell you how that happened. Woody Allen says half of life is showing up. I was on the panel. Harold Varmus, who was not particularly inclined to clinical research when he started, had put this panel together, and I was on the education committee with Gene Wilson. So we had a meeting in August at the NIH. Now, the NIH in August, they're building one, not air-conditioned, I mean, it's hot, sticky. So there's a huge committee. I come into the room, there's Gene Wilson with a yellow legal pad and me. No one else shows up. There's about 25 people on the committee, August. Blew it off, So Gene said, okay, Bill. Opens up the pad, he said, what do you want? And I said, let's have a 5-year award for young investigators right after a training grant. So we wrote that down. Let's have a mid-career scientist award that would last 10 years. Wrote it down. Let's give loan repayments for the people on this. Wrote it down. And let's have training programs for the people, okay? Now, we didn't have any Ks or anything. So we wrote all those things down. We costed them out. And that day, the Education Committee passed it unanimously. The 23 people who didn't come all said, sounds good to me, went off to Varmus, who then took it to the Institute directors. And to this day, actually, Steve, the head of the Arthritis Foundation, thinks these are their awards because they put the K23, K24, K30 designation on. I think there's no limit to what you can get done if you don't care who gets the credit for it. So all these programs went in, and Gene and I still chuckle about this. It's half a life is showing up. Woody Allen's right. The third thing is society's impatience about the poor drug approval record, the doubling of the NIH budget, and the accountability. The drums of accountability beat heavy inside the Beltway. We don't quite hear it as much, but if you're at the NIH, and the NIH directors hear this a lot, and particularly the NIH, the director, but the Institute directors hear it too, and the skepticism is growing. And in fact, there's been articles written about the myth of the biotechnology revolution by these two guys in England. And they went through and looked at all the literature on patents, new molecular entities, a variety of things, and looked at all the objective data and said there's no empirical evidence to support the notion there's been a biotechnology revolution. It's only shifted the innovation bottleneck to the target validation and clinical evaluation, where a much more complex process is involved and costly. As a consequence, biologic knowledge in the laboratory is not being translated. This is the block. This is the first translational block we wrote about. And the policymakers need to move away from this increasingly discredited linear model, that's the Bush model, of innovation that sees new drug and diagnostic as little more than the application of basic research. Then the IOM's Clinical Research Roundtable came up with 4 years of deliberation and put names on this, which is the names people use, the 2 translational blocks and the National Clinical Research Enterprise. And we'll talk about the 2 translational blocks, but the enterprise said we're all in this together. Each place has viewed this as a silo. And in fact, when you begin to look at it for 4 years with everybody involved in the healthcare system, payers, providers, insurers, etc., there really is an enterprise here. Except the enterprise has no way of communicating silo to silo. And CTSAs could be one of the ways to do that. So now if you look at the bench to bedside, this is the first translational block. This is where Albright operated. This is the innovation piece, and this happens exclusively in academic centers and at the NIH. Biotech actually has picked it up, but that's because they're the first derivative of academic centers and usually driven by academicians, actually, who set up their own companies. And I actually declared in the opening slide that I have one with Bob Langer. And the dollars, however, proportionally come from the NIH. The Genome Project is reshaping the way this is done by moving from monogenic to polygenic, looking at populations and phenotypers, the things that you deal with every day. But it draws a sharp line between a clinician and a clinical investigator. And that's not, unless you're working in the field, that's not obvious to you. But these large databases and phenotyping and the ability you get from the longitudinal medical record now is first derivative of that, can be really the phenotyping, and they can image things. So it's quite clear that these are the type of things that we work at as academic centers. Where we've not been so good, and actually Nancy Bennett will talk about this afternoon, is this second translational block. If you just take aspirin, beta blockers, and ACE inhibitors, 3 things all shown to save lives in randomized large clinical trials, and look at populations as to who's using them, who've had an MI, this popped up by the pharmaceutical company as a shortfall of marketing. In other words, there's 40 million people who've had MIs. They all ought to be on these things. No more than 60% are on any one of those three things. So we're not getting this out to the community, and that's the implementation piece. And this is a tough one, as we were talking before. The dollars typically come from industry, public health, and a little bit from AHRQ. The payers are increasingly interested in this and writing this into the contract. So we have pay-for-performance contracts in which we have to make sure this is done, and we negotiate that into the contracts because it helps our CEO drive internal processes and synchronize them with payers. It's actually one of the regulatory pieces to pay attention to that does work well, actually, as you need a stick in every... Carrots aren't enough, let me put it that way. These are population-based considerations, though, and they're the largest gap in public health. But it forces clinical research to integrate with delivery of health care, with all of the problems of equality, access, portability. All of those issues are stuff that they're tough problems. It comes up as pharma shortfall and under-prescribing, and this led to direct-to-consumer advertising with all of its evils, but that's what gave rise to it. There is still an under-prescription problem, even though there's an abuse of prescriptions, and I see this with testosterone prescriptions are going crazy for things that isn't necessary, but still the beta blockers aren't being prescribed. So it hasn't addressed the problem. So the other thing is the NIH roadmap, the re-engineering and the CTSA that you're here to celebrate, while still a work in progress, clearly is one of the potential tools. So what's new here? Well, I think we're moving to a cyclic model of research, which is the roadmap for biology of the 21st century. And this is one of the websites we have for our genetics center, but I think it's got the description pretty well. We start at the top with patients and families, interestingly. We describe their phenotypes, a difficult and laborious piece. We use those through genetic or omic tools to identify the underlying genes, and then you drive it into the basic laboratory, having identified it. There the refinements occur, and the critical nature of the importance of the investments in basic research emerge as new diagnostics and therapeutics. And if you have this right, you bring it back to those families and it works. If it doesn't, you have genetic heterogeneity and you go around the cycle again. So this is, from a reproductive endocrinologist point of view, it's a regenerative cycle. It's not a sterile linear cycle, so I like that. But the other piece of this is, I've seen this now come from the genetic heterogeneity to let you know about the pathways when things don't work. So this capitalizes on, I would say centralizes, patients and disease in the orientation of this. These are convergences which only you have. This is our moment. If we don't realize this and capitalize on this, drive pharma to this, drive the NIH to this, we've lost the golden moment here in academic centers. This is a self-reinforcing, non-linear cycle. It's generated from a reproductive point of view. Its weakest links, however, are the slow translations into the human, the lack of reliable models, and the fact that there's no infrastructure, and the CTSCs are in part ability to develop this. It has typically lacked the space or systematic access to the basic science, project management, and all those things. Again, something that the CTSA should be able to do, make more efficient. And the model problem is there isn't any prior link between diagnostics and therapeutics at such early level discovery, but I think the cyclic model changes that, actually. And I think the integration and convergence of all this, the imaging, molecular markers, are the things that we really have to pay attention to. And the omic technologies that are being driven by these and the engagement of the IT and bioengineering communities are not to be missed opportunities. Okay, let's run this through now, your center, and say, how has this changed those investments? I won't pay as much attention to the NIH, although they're trying to rebalance clinical research, and they've really got a central problem, their lack of their patient base, their inability to integrate with academic centers. All the great headaches you have become your assets here in this involvement. And the idea of the fact that the yellow berets are gone and the intramural talent pool is very different than it was 30 years ago, and their difficulty in addressing system affiliations. Let's ignore those for the moment, because that's the NIH's problem. We're not here to solve their problem. Medical schools now become the home for clinical research. It's obliged by the CTSA. The dean is here. I mean, the dean wouldn't, well, you got an unusual dean, he would have been here, but I mean, in most places, the dean would not be at this, because clinical research was not being decided by the dean's level. Suddenly it appeared on his or her desk when the GCRC was in danger, you're going to lose $3 or $4 million. You better pay attention to that. So it erupted, and we saw this at the forum where all the deans were here and suddenly want to know, well, how are you going to handle this problem? The worry now is, are they going to redelegate it down to the GCRC-like mentality? Because the people who ran the GCRCs never had the resources or the mandate to think about the system. It was to make the local trains run on time. The mandate is bigger here. It has to stay on the dean's desk, and we're redefining who's a good academic center or not, and I think there's a lot less than we think. I think one of those definitions would be the deans that continue to pay attention to that. In addition, revising promotion, changing education, putting in masters, devoting space to these. These are all the mechanical issues that are being done. Also the partnerships, the places that, formerly the idea of having an affiliation with a medical healthcare system, you didn't know if you wanted it. In fact, University of Chicago was trying to sell off their hospital. The president was trying to sell off the hospital at one point because it looked like a black hole into which you could sink a lot of money, Now those become major assets. The clinical research cores, moving them out into the hospitals and into the community. It's clear that the PhD's discovery model become very important, and their new tools become fueling the engines of this if they can work with the academic centers. And the clinical researchers are important, their training programs and their translation. And the prediction is they're going to start to get big recruitment packages, and I'm seeing that at our hospital. I just had a young clinical investigator leave for $1 million package, a translational investigator to another institution. I haven't seen that in my lifetime, but it is one of the logical consequences that would happen. They're going to be competed for, they're an under-resourced, their market value will rise, and people will get it or not get it, and people will put the steel on these people early on. And in our institution, which is a soft money institution, when somebody comes along and offers you $1 million of hard money, you pay attention. It's clear that the medical schools must become the home, but they're all ill-equipped to do that. And is this CTSA going to be just an added layer, or is it going to be a catalytic layer? Not clear. It's clear that within the academic centers, the genotype-driven proof of concept studies using genotyping and targeting pharmaceuticals are the way to go in the future. It's been resisted heavily by pharma because it makes big markets into small markets. So you want everybody with hypertension to try the drug and only 40% work. Trouble is, it takes a long time to get them approved. If you go in with 40% of people, 100% responses, it goes through the FDA like that. And their most expensive thing they have in their books is an unapproved drug because it's in clinical trials and it's eating cash like you wouldn't believe. But the idea is the marketing people can't drive the pharmaceutical companies anymore. The scientists have to. That's their fundamental problem. The marketing people drove pharma and the scientists lost, and that's got to be reversed. And the tools are there to reverse it. It's also clear that the medical schools have now discovered public health. They actually can spell epidemiology and decided it's not an epididymis. And in fact, they actually now have departments for this, and they attach the schools of public health and have to integrate them into CCSC. They use websites. They're tremendous. Harvard's Medical School's website is the most heavily populated website for patients who want to find new therapies. And networks, of course, are what the academic health centers are about. It is our moment to be a translational engine of the 21st century or not. And that is the choice that is in front of all academic centers, you among them. So what are the three lasting policy implications? We're back to the hospital model. Basic scientists are coming here. The mutations are here. The patient populations are here. The opportunities. So the advice to young physicians, stick around. Stay close. That's where the money is. Seek these basic research partners, and the CTSC should be the catalyst to be able to make these. There's a new biology out there with proteomes. The medical schools have to follow suit. They have to change their models. Teams have to be rewarded. Promotions have to be accordingly distributed, and space has to go that way. Industry will eventually get it, and if we're prepared, they will come. Clinical researchers will be competed for, as I told you before, and it's happening. But we have to retain the emphasis on recruiting the best and brightest at this particular moment because the opportunities are unparalleled. And it has big infrastructure implications. We've got to really subsidize all the cores. We have to make them work together. We can't view the clinical trials as a profit center anymore, and pharma just doesn't build lasting infrastructure. We have to recognize that. And the real opportunity here are the CTSCs, which you've been lucky enough to recruit. So the good news is that academic centers have made huge investments if you look at this in science, or you look at it through the 15 to 25 billion dollars of new centers have been built in academic centers. As I go around the country to give this lecture at various places, it's amazing what building has gone on. Part of that being the NIH doubling, and we're going to have a little short sheet at the other end, I can see. But in fact, they have been built, and philanthropy has been driving them. Now, we're in for some tough time for that. It's also clear, though, that the patient disease model availability in the genomic era is expanding. Actually, I'm on a science sabbatical for 6 months, and I'm spending half of it at the Broad and half of it in our genetics center because that's where our own research is going. But the types of data, the ability to search large amounts of data, the research patient data registry we have, and the building of other infrastructures like biorepositories, tumor registries, core labs, that's where we have to put a little bit of our money. The tradition of physician scientists at medical schools is strong. We've always had good people. We've always had good training programs, so our supply side is strong. And the finances, until a month ago, were actually pretty strong, and I think will be strong again. But philanthropy is one of the pistons of academic centers that depends on, and it's a down piston for the moment, but it will come back. So when the day is done, these two men have had a big impact on medicine. I would say that Albright was right for the 30s, Bush was right for the past 60 years, but we're now coming into a new equilibrium in which the bedside and bench can partner in an even way, and in fact, this is the opportunity of the moment. So for the young people, the final thought, a few final thoughts. It's the best of times. The tools are tremendously powerful. They're game changers. Our patients can teach us amazing biology. I used to call patients unique biologic opportunities. So UBOs, pay attention to UBOs, the ones that are different. It's the variants in humans' diseases that should point that out to you. The opportunity to alleviate human suffering, which is, after all, the core of why we're doing this, at least why I'm doing it and why most people are doing it, is truly unique. And that's the thing to focus on through thick and thin resources. The ability to alleviate human suffering is what we should be about. Clinical research is really in for a renaissance in the CTSA era, and the best is yet to come. Thank you for your attention.